Hi there, everyone. So, just saying that for lab, we're going to be doing some osmotic stress and antibiotic resistance today, and we'll see about our unknowns as we go further down the road. The exam is going to be dropped on Friday, and it'll be due on Monday, and that's all I have to say about that. On Moodle, there is the chapter by chapter review things to know sheet, so go through that and look at that. And that's about all I have to say about that. All right? Good. So, we've been covering viruses sort of by different systems in terms of where they attack. And we're now going to shift from the respiratory system into viruses of the circulatory and nervous system. And so I guess it wasn't all respiratory system. It was also nothing but it was respiratory and dermal. So we were covering things like chickenpox and HPV, the human papilloma virus, as well as the flu virus and a little bit of coronaviruses as they stand now. We're going to shift now to talk about different systems that are invaded by viruses, specifically looking at the circulatory system and the nervous system. Herpes could kind of get put into the nervous system as well, but we're going to have a little bit more of a distinction here. And one of the first viruses that we want to get into that is involved in the circulatory system and specifically affecting blood is the virus that causes AIDS. This is the human immunodeficiency virus, and it causes a disease known as the acquired immune deficiency syndrome. It is a classic example of Koch's postulates in that the first thing that had to be done, is recognizing a common disease, and then second, purifying the disease-causing agent. So the first reports to the CDC happened in '81 from San Francisco, Los Angeles, New York City, and specifically, one of the things I remember reading about at this time is that the CDC was recognizing some very rare pathologies. Things like oral candidiasis, which is a yeast infection in the mouth, cytomegaloviral pneumonia. More importantly, I think, is Kaposi sarcoma, which was something that was so phenomenally rare. We looked at it in the dermal virus section previously, but the CDC apparently had seen like 2 or 3 cases in its entire history. And now all of a sudden, all these otherwise healthy young men from San Francisco and Los Angeles were starting to show up with Kaposi sarcoma. So there were other kinds of bacterial infections as well. Just the point here with Kaposi's, this is a virus that causes the sarcoma. It is a kind of cancer that our immune system is brilliant at taking out. So a healthy immune system, you never see this. But what we have here is a virus that is beginning to attack the immune system. So the virus is identified in '83 and becomes named in '86. The actual discovery or purification of the virus is one that ended up having a fair amount of political drama with it. There was 2 different groups, Robert Gallo at the NIH and I think Montague, a guy in France. Both purified the virus. And there was accusations that one had not purified the correct virus. All kinds of things going on back and forth. There was even a third guy named Peter Duesberg in Berkeley who was so convinced that his arch enemy Robert Gallo had not purified the actual virus that he said that he would be willing to be actually injected with it, because that's sort of the closing link in Koch's postulate is you purify the infectious agent reinfect something and see if you have the same exact symptoms. And it's probably a good thing that he didn't allow himself to become infected or be injected by it, because, no, they did have the virus. One of the thoughts was this might be a virus, because it was starting to get to be a very significant issue, that would have a rather lucrative vaccine opportunity. And in part because the virus actually damages the immune system, it's been very difficult to make a vaccine against HIV. We'll talk about that a little bit later. All right, so the cell type that it binds to and infects are a set of white blood cells known as T lymphocytes. And these are also known as helper T cells. And they're a critical part of the immune system. So, we talked a little bit about this before, that viruses have to have a protein on the cell surface they can bind to. That receptor is what is going to allow the viral nucleic acid to actually enter into the cell, or the virus itself to enter the cell. And unless that can happen, the virus is irrelevant. You don't care. The virus that causes AIDS binds to a protein on all T cells, and what's known as the T4 receptor. And this is why HIV infects this specific cell type. So HIV infection is still an issue in parts of sub-Saharan Africa. The U.S. is pretty low. Parts of China, the Soviet Union, end up having, I guess it's the Soviet Union, have higher rates still. Africa has got some of the highest rates, though. So it is increasing in parts of the world still. Now, one of the things is that it used to be absolutely lethal, and we now have a set of antiviral drugs, and in part, study on the virus that causes AIDS, study on HIV, has really advanced a lot of viral disease drug therapy. So, the primary infection, a low-grade fever, mild flu-like symptoms, probably something you might not even notice. There then is a fairly long latent period. The virus is still being continued to be produced, and then eventually this turns into full-blown AIDS. So we've got the virus, HIV, and then the disease that manifests itself is the acquired immune deficiency syndrome. And this is identified because the T helper cell count drops. And by looking at the T cell number, that's how one of the ways they were back then able to diagnose HIV. It took a while for an HIV test to be developed. And because HIV is transmitted primarily through blood, there was a time back in the early mid-'80s where the Red Cross blood supply was in fact contaminated with blood that contained HIV. And it was a pretty serious tragedy in that for a while there were people who would have blood transfusions actually contract HIV through contaminated blood. So now, I'm not sure, has everyone here donated blood? Anybody not donated blood? I haven't donated blood in a while, but there's a lot of questions that you are asked. And part of what the Red Cross is doing is kind of asking you in a somewhat obtuse way, what is your risk factor for potentially having HIV? And there are things that are contributing to risk factors like IV drug use, aggressive sexual practices, and they ask you a set of questions. And now, we were talking about prion diseases, mad cow disease and Creutzfeldt-Jakob. There's been a new set of questions about, have you traveled to Europe in the past 6 months? And if you've lived in Europe for longer than, I don't know, a year or something, they pretty much are not going to use your blood in transfusions. And that it was because the possibility of a Creutzfeldt-Jakob outbreak in Europe that probably hasn't really manifested itself. But there's these questions that they're asking, rather than saying, "Hey, do you think you have HIV?" or "When was your last HIV test?" Because we don't typically test people. They're asking about risky behaviors, and then of course at the end there's a little sticker where you have the final say. Pull this one to say yes, I think my blood's good. Pull this one to say, go ahead, take my blood, so nobody says anything. But my people around me don't say, "Hey, why aren't you donating?" But maybe either test it or use it for other blood products or for other systems, but don't use it in transfusions. All right, so HIV is a retrovirus. That means it is an RNA virus. It initially attaches to the CD4 receptor on the T cell. It then uncoats and dumps the RNA into the cytoplasm. The RNA requires the conversion into DNA using reverse transcription. And reverse transcriptase is one of the proteins that the RNA can code for. The DNA gets into the cell, and the DNA actually can integrate into the genome, into the host genome, so it can lysogenize. And now the DNA of the host cell is in fact the spot where the viral RNA can be generated. And so the viral RNA is being generated, the RNA gets out to the cytoplasm, viral proteins are being produced, the infectious viral particle is being generated and loaded up with RNA, and we have budding release of the mature virus. Now, it doesn't end up killing this cell, at least not right away. This cell becomes a viral producing factory. So that makes it a little bit difficult to treat. Now, HIV has been able to survive a whole range of different kinds of immunological assaults. Our immune system, as we've been mentioning, is your number one line of defense against viruses. We don't have great antiviral therapies the way we have antibiotics. These are antibacterials. So one of the things that happens is that the virus is able to change its coat protein and avoid immune response by essentially carrying out mutation that results in a few viruses escaping and then continuing the infection. So you have the original infection, the immune response may be able to take out some of it, but not all of it. And as you have these mutations picking up, you end up with the virus continuing to propagate. And one of the things that happens is that mutations generally are going to be deleterious, but the virus produces so many offspring that even though most of them may be non-functional, a few are, and they're able to actually infect and continue on. So they're able to begin killing the infected T cells, and then there's other T cells that are not dead but producing more viruses. So yes, there are some drug treatments, and in fact, if you keep up with HIV politics and things, starting around the late 80s, this drug cocktail begins to be prescribed, and this is a set of drugs that has anybody ever seen the movie or the play Rent? No one. Okay. It's about New York in the mid 80s, late 80s, and there's a little scene there where somebody's alarm goes off and two people look at each other and go, "Oh, you too?" Because in order to take the retroviral drug therapy cocktail, you had to have a real specific schedule because some of the drugs couldn't be taken together, and you had to have a very specific timing and spacing out of these 3 different drugs you were taking every day. And that makes it difficult for people who don't have maybe a particularly stable lifestyle to be able to maintain this. And so the therapy is a lot simpler now, and it works pretty effectively. There are people that are living with HIV infection, and because of the success of the retroviral drug therapy, their viral count is essentially 0, but there are cells that still have the virus, latent cells, that were you to go off the drug therapy, the virus starts to come back up. So a couple of the antivirals. There are protease inhibitors which are able to block the pre-protein, the RNA that's being generated, or the RNA generates this initial big protein that has to be broken up by a protease, which is actually part of the pre-protein, like a snake kind of chewing on itself. Protease inhibitors are a good place to start. Another thing that you can do is have a nucleotide that looks like a standard nucleotide, but is missing one of the hydroxyl groups to allow for the DNA to be continue to be polymerized, and the enzyme reverse transcriptase ends up actually being less careful about what nucleotides it picks up. Our own DNA polymerase or RNA polymerase is pretty careful in that these nucleoside analogs don't get picked up very well. And so you take this, it's going to affect the reverse transcriptase, but it's not going to affect your own DNA polymerization. So it works fairly well. And then there are drugs that are blocking the integration of the virus into the genome. And this allows for essentially a halt to viral production. You're still infected, but you're not producing any more virus. So one of the questions that was initially pursued, and is now very similar to what we're doing with the coronavirus, is where did this virus come from? Why now? How did it happen? And much like the coronavirus, increased interaction with humans and parts of the environment that we had not been in. As you start to clear more forest, you start to interact with organisms that we have never been interacting with at a level that we've never been interacting with them. Chimpanzees, which were a source of one of the first HIV strains, were being consumed. This is bushmeat, as it might be said. What is a sooty mangabe? I don't know, but it apparently was involved in animal trade. And then this is how the initial infection makes its way into the human population. There's actually signs that HIV had been around for a very long time, but the expansion of it within the human population comes about through different kinds of behaviors. Now, next, once it's in the population, it gets spread in very specific spots. Prostitution is one of the main causes for spread in Africa. And additionally, as people begin to invade the jungles for food or to expand their property, you get exposed to more strange zoonotic species, these different kinds of organisms and things. Additionally, sexual practices change, and that changes the way the disease is spread as well. So, herpes, again, you hear that, YouTube? Yes, herpes. If you check our comment section, that's what we get comments on. There are multiple herpes viruses. I mean, we talk about the fact that we categorize viruses by some fairly primitive, at least from terms of speciation, primitive ways. And we lump them together. While they're different, distinctly different viruses, we have a class of viruses that we think of as herpes viruses. So, beta herpes virus, these are actually involved in T lymphocyte infection. One kind of herpes virus known as CMV, or cytomegalovirus, is one of the common causes of mononucleosis. The gamma herpes virus, these are also infecting cells in the bloodstream. And one of the types of herpes is the causative agent of Epstein-Barr syndrome, which is, it's linked with leukemias, also linked with chronic fatigue syndrome, and it's interacting with the B lymphocytes. Kaposi's is another one of these that is affecting some of the blood cells as well. So these are herpes-like viruses. Another class of viruses that you perhaps have heard of is the hepatitis virus. And these are a very diverse range of viruses. Their only real similarity or commonality is the fact they infect liver. And so this is, again, one of those places where we have a whole range of organisms, viruses, that are labeled the same because they are affecting the hepatic system, the liver system. And so we have a range of hepatitis viruses, but they actually are fairly distinct viruses. So there's Hep A. This is an RNA virus, a little bit like polio, and the virus causes a common cold. Pass through the oral fecal route, oh boy. Hep C is a enveloped virus, an RNA virus. It is similar in its passage and operation to Hep B. And Hep B here is a DNA virus, so that means it's a complete different mechanism of operation. This is able to become acute and then turn into a chronic liver disease. And Hep D is sort of an odd one. It contains actually HBV proteins, and it's actually part of a co-infection. You have to have HBV, Hep B, in order to have Hep D floating around in you. So hepatitis B is one of the newer and more dangerous hepatitis viruses around. So the virus is brought in through the mechanism of endocytosis. That is, the entire thing is brought in as a membrane. And one of the things I think to take away from this is, as we've been looking at these different viruses, the mechanism by which the virus gets inside the cell is as diverse as the viruses themselves. So we have some viruses that are binding their membrane, fusing it to the cell surface, dumping their contents into the cell. Other viruses, like this one, actually get engulfed into the cell. And once the virus has been engulfed, the core is released. It is something known as uncoding, as the virus is opened up and the genome is now revealed. The genome gets into the nucleus, and it's actually able to integrate into the genome. And from there, we have a sort of a circulation of this material back and forth. The transcripts can go out. You have the RNA being converted back into DNA. The genome then is placed into this component of the cell called the Golgi system, which is responsible for a lot of membrane packaging and proteins being placed into the membrane. And you end up then having this packaged virus being shot back out, carrying the membrane. So again, not going to be a way of, it's not going to immediately kill the cell. So HBV is a serious problem in areas of high needle drug use. So if you're going to do needle drugs, make sure you don't share your works, number one. Probably a good idea to use a fresh needle if you can, and if not, washing your needles out and with Clorox. But I'm not sure if you've ever been doing injecting, but a fresh needle is a lot nicer than even a once-used needle. Let's not share needles. I guess actually let's not do needle drugs. It's probably a better thing, but you know. So it gets into the blood, and from there it makes its way to the liver, where it's actually able to integrate. And you have a couple of different outcomes. You might be able to knock it back, or you have an acute infection, which is going to hopefully be knocked back. Otherwise, this chronic infection happens. The virus continues, and you have long-term inflammation, and that's going to lead to liver damage like cirrhosis and or cancers over time. we're going to stop here. Well, it was a bit of an abbreviated lecture. I'm not flying through this. So, there you go. Thank you.